master's research with Evgeny and with Brian Hunt as well in September. And I'll be looking at the prey diet of juvenile sockeye salmon across spatial and temporal scales. I'm also in the early stages of my research, so I'll just share with you a few things today. First of all, why we all love salmon, the Salmon Early Marine Survival Program that this work is a part of, and then touch briefly on my own research. So Pacific salmon have tremendous value ecologically, politically, culturally, and economically. They carry nutrients between freshwater and marine environments. They provide food for larger predators, including us. They support large commercial and recreational fisheries, and they're a cultural icon to many First Nations communities in our province. Of particular interest are sockeye salmon. However, several stocks have been in decline since the late 1980s. Of particular concern are the stocks in the Fraser River. The average returns in the early 1900s, you'd see about 40 million sockeye returning to the Fraser. If you look at last year's returns, it was about 900,000, which is one of the lowest returns in recorded history. So there's something going on. We're not sure what it is. There's a lot of research being done on sockeye salmon right now, and one of the theories on one of the factors causing this decline could be mortality during the early marine phase. So there's a few groups who are now doing research on this, one of them being the Salmon Early Marine Survival Program. This is a collaborative project between Sockeye Institute, Salmon Coast Field Station, University of Toronto, and UBC. It started in 2015, and I joined the program last summer. And the goal is to really shed some light on what is happening during the early marine phase. What factors are affecting early marine survival? So our program focuses on Discovery Islands and Johnstone Strait. So here we have a map of the Fraser River watershed and potential outmigration routes of sockeye salmon. And you can see our work is concentrated in the red box there. And if we zoom in, these are our study sites. We have the program is split into two different components. So we have a crew based on Quadra Island here in the middle that covers the Discovery Islands, and we have another crew based up on the Salmon Coast Field Station in the Broughton Archipelago, and they cover the Johnstone Strait routes. So as you can see, we have a very wide spatial coverage that covers the major migration routes. And that's what's really unique about this project, is the high spatial as well as temporal resolution. We go out there early May, and we are catching sockeye and other salmon throughout the migration period to early July. And from the fish we catch, we look at everything you could look at. We're doing genomic profiling, we're doing stable isotope analyses, as well as fatty acids, to shed some light on some of the food web dynamics. We're looking at stomach content analyses, otolith microchemistry, scale growth rings, as well as doing genetic stock ID on a subsample of our catch, and looking at sea lice loading. But my particular interest and my involvement on this project is looking at the stomach contents. So using this amazing data set, here's the question I'm hoping to answer, and that is, how do the feeding conditions vary, both spatially and temporally, across the juvenile sockeye salmon migration through the Discovery Islands and Johnstone Strait? And again, what's really unique about this is being able to look at both the spatial and temporal components with such a great level of detail. So here's how I kind of break this down. I'm going to be looking at what's in the water versus what's in their stomachs. And this is in terms of their prey. So for sockeye, that's usually zooplankton. And for those, I'll be looking at the diversity, the relative abundance, the size distributions of the zooplankton of the prey, as well as the stomach fullness and prey selectivity of the sockeye salmon. And I'll be doing this for the Discovery Islands and Johnstone Strait over space and time. So some of the questions we want to know is, how does diversity change in these different areas? Is it different in the Discovery Islands sort of northern strait of Georgia, where we see more thermally stratified waters versus the very highly tidally mixed Johnson Strait? Are we seeing different size distributions across similar gradients? Is there, more food available in thermally stratified waters versus in the tidally mixed waters that might cause a higher occurrence of empty stomachs? Are sockeye being selective towards certain zooplankton in their diet? And does that change as they grow? Does it change along their migration? So there's a lot of questions to be asked here and still a lot to learn. And so I'm hoping that from my research, I'll be able to shed some light on one of these factors influencing the early marine survival, which is their diet, as well as contributing to this growing body of research that's occurring right now, with lots of research groups starting to look at the early marine phase and the factors that influence it. And particularly when paired with genetics data, this could shed some light on specific stocks and how they're sensitive to changes in their diet due to their life histories, due to their migration routes, and the timing of their migration. So that's it. That's just a very brief overview. If you have any questions, I think we have time for one or two.